Okay, so a couple of terms that we'll use over and over again. And one of them is passaging and splitting cells. So what does passaging or splitting cells mean? So let's say I have a dish of cells. So typically we call it a petri dish, but we don't use the word petri in cell biology. So we're just going to call it a dish of cells. And I have my cells. These are epithelial cells. And what they will do is they will be attached to the bottom of the plate like this. Now, typically what will happen is they'll start out, and we're drawing this in 2D, but you can imagine in 3 dimensions what that will look like. And there will be some gaps in between them. And of course, these cells are attached to the bottom of the plate by proteins that they have. So kind of like how they attach to the basement membrane in tissue. So epithelial cells, one of their requirements is that they must be attached to something to not die. When they're attached, they're getting signals going into the cell telling it, don't kill yourself. Don't go through programmed cell death. Yes, this is a thing. Now, there's only so much surface area on the bottom of a plate. And when these cells go through mitosis, they start to fill up the bottom of the plate. So here we go. We're going to fill these gaps in. So we have filled these gaps in, and from this point on, I'm not going to draw the little green proteins attaching the cells to the bottom of the plate. Just know that they're there. Okay, so now that the whole bottom of the plate is full, basically the cells have nowhere else to go. They can't grow anymore. They're out of space. And when they're out of space, they become somewhat metabolically less active than they were before. Now, in the case of some cells that are used in a laboratory, once you get to the state where there's no more room to go, eventually after a couple of days, the cells will actually die. So it's not good to have the whole bottom of the plate covered with cells. And when the whole bottom of the plate is covered with cells, we say it's 100% confluent. It's this idea of confluency. We say it's 100% confluent. You don't want to reach 100% confluency. The reason why, besides the cells might die, is that when you're getting up to 80% or 75% confluency, metabolic activity starts to go down. So imagine that I have a range, 100% confluence, and you can't have 0% confluent. That means you have no cells. So let's go with 20%. And we're going to have another measure here, which basically is metabolic activity. So metabolic activity, that just means that the cell is using ATP, building its proteins. It's very active because it has to go through cell division. It has rooms to divide. So cell division, there's a lot of things going on. You have the G1 and the G2 where the cell is making a bunch of proteins to get ready to grow and etc., right? You can imagine that metabolic activity is very low here, and somewhere around here it's going to be in the middle high. Once you get to very low confluency, it starts to get low again. 25% is fine. You're still going to be sort of in the high range. At below 20%, meaning that you put so few cells in, the amount of metabolic activity is going to be low. The cells like to be around other cells, at least have other cells close by to them, typically. Now, why is this important? If I'm doing an experiment, right, I want to be able to repeat that experiment and get the same results. I want somebody else to also be able to do that experiment and get the same results. You have to be able to repeat an experiment multiple times and get the same results to know that, hey, whatever I'm seeing is correct. Whenever you do an experiment, you want to keep as many parameters as possible constant while changing one parameter, maybe 2. The more parameters that are variable, the harder it becomes to actually see what's happening, to come to any conclusion. So one thing you want to control is metabolic activity of your cells. So typically, people will always do their experiments in the area where you have this high metabolic activity. You do not want to be in these zones. You definitely don't want to be varying around high confluency and very low confluency and then in the middle. Because then this whole thing of metabolic activity becomes a variable in your experiment. So you're very careful to make sure that, okay, I am going to have all my experiments. If I'm really, maybe I am looking at the phenomena of what happens at higher confluency. Like what happens when cells are very close and bunched up together? Well, then I'm going to work in this area, right? But typically, if you're just doing something where you want your cells to be metabolically active, you make a decision that every little experiment that I do in this study will be around this area, right? You have to make that decision because you don't want that to be a variable in your experiments, because then your results could be all over the place. But how do you get from 100% confluent to 20% confluency? So I'm going to erase this stuff so that we can continue on. So again, I'm not going to draw the little green proteins showing the attachment of the cells to the bottom of the plate. So what I do is, and what all cell biologists do, let me scroll up a little bit. You go ahead and you take these cells that are in this plate here, right? And you divide them into multiple other plates. So let's say we're going to divide them into 3 plates. And I think I can fit it into here and keep the size somewhat similar of the plates, right? And what will happen is that now these cells are on the bottom of the plate and they have a little bit more room. You went from 100% to maybe, actually let me take one away, to about 30%, right, on each. Now this action is called splitting. Splitting cells. So what we do to keep track of how old our cell culture is you keep track of the split count. Meaning that, okay, if I was at split count 0 here, now I'm at split count 1 here. Meaning I've split these cells 1 time. So splitting is also called passaging. So we say that, okay, cells were at passage 0 here. So 0p, and that they're at passage 1. So if I were going to split them again, or passage again, then the next one would be passage 2, and etc. By keeping track of the passage number of the cells that you're using, you kind of have an idea of how old your cells are. Why is this important? We talked about genetic instability. Most cells that are used in a laboratory, a large proportion, are either of cancer origin or have been transformed, and they can be maintained indefinitely, meaning that I can keep passaging them and go to passage 100, 200, 300, whatever I want. But there is the problem of genomic or genetic instability. And how genetic instability works, how you start to get mutations, it happens during the S phase of mitosis. Every time a cell goes through mitosis, at each S phase, it will pick up some mutation. Sometimes the mutations can be dramatic, sometimes it can be very benign, but it will pick up mutations and it will start to accumulate these mutations. So eventually, as you have many passages, as your passage number increases, that genomic instability starts to accumulate and the phenotype of the cell will change. Now, how do you know if the phenotype has changed? And this is why a positive control is always important in your experiments. So I'm going to make an example. I'm a virologist, and I'm going to use the example of virology to get this point across. Let's say that I am studying a virus. So here's my virus. Of course, a virus by itself doesn't do anything. It needs a cellular host to actually do something. So what I do is I take my virus, I put it onto a plate of cells, I infect those cells, right? Here's my cells. I infect those cells with the virus, and once these cells become infected, so we'll just shade them green for being infected, they start to produce more virus, right? And I can take naive cells, meaning cells that haven't been infected, treat them in some way, and then try to infect them and see if the virus infects better or infects worse, meaning there's less infectivity or less ability of the virus to make more of itself once it infects a cell, and etc., right? Different experiments on the virology of this virus, the replication cycle of this virus. All my positive control is doing is that I infect the cells and virus comes out, and I keep track of the amount of virus coming out. And let's say that I start out with passage 25 cells. I keep doing the same positive control with the passage number increasing. So I just keep doing it, let's say to passage 100. Okay? And eventually I'll get to a point where the virus can no longer infect the cell. So let's say I get to passage 100. And here's my cells, my passage 100 cells. I put the virus on, and actually it might infect. I put it on, but now the output virus that I get from these infected cells is lower than my positive control. So it's much lower. So at the point where my positive control stops working, or stops working at the level that it was working before, that means that I have a range within those passage numbers. Whether it's, let's say it works normally from passage 25 to passage 40. That means that from passage 25 to passage 40, this system is working fine. After passage 40, the phenotype of the cells change, and they're not getting infected at the same level they were before. So any experiment I do on cells from passage 40 and below, I'm actually doing it in a cell population that is different from the cell population that is a lower passage number than passage 40. So I will make sure that I do all my experiments between passage 25 and 40. Actually, I might walk it back and just go from 25 to 38 or 35 and not even get close to 40. That way, I ensure that whatever experiment I do is repeatable. Okay, guys, we'll stop there and we'll continue later.